There we go. There we go. That looks more like it. Yeah, I went backwards a bit. So this is the ATCase that we were talking about, and we had left off on the binding of the CTP to those regulatory sites, like so. That's the CTP being bound to the regulatory sites. Remember, this is that feedback inhibition, right? Okay. Now, this right here is the T state. This is the less active state. This is the state that's going to be favored by binding CTP. The R state, on the other hand, is favored by substrate binding, and this is the more active state. The increase from L is from 200 to 1,250. And so this is one of the reasons why I'm always using active, less active, more active state, rather than inactive. Notice that difference in terminology? Is very rarely does it actually fully deactivate an enzyme. Okay, when you're doing an allosteric interaction. Substrate binding will fully deactivate an enzyme if you modify the binding site, the actual active site, right? But in this case, we only go from 200 to 1,250. Now keep in mind that that actually is a very large difference, right? Okay. Anybody have any questions about this? Okay. Yes, yes. That's essentially the ratio of activity of the enzyme. Okay. Okay, so the CTP is going to increase the initial phase of your sigmoidal curve. This is where we see that this one right here is actually adding 2 micromolar of ATP, and we can actually see that the addition of ATP is actually an allosteric activator. Can you see how it reaches that curve faster? Okay. And so the CTP, on the other hand, is an inhibitor, right? Allosteric inhibitor. And so what you see here is that moves it downwards and to the right. This is going to increase that initial phase that we would have of our sigmoidal curve, i.e. it happens for longer on the initial phase, and it takes longer to reach the max, right? Okay, so ATP, again, an allosteric activator of the ATCase. This can bind at the same site as CTP. This, instead of CTP's favoring the T state, it will instead favor the R state, which is why it's an activator. It's going to increase your net enzyme activity by favoring that R state. The heterotropic effects is going to be the effect of non-substrate molecules on allosteric enzymes. It's sort of, think about it as one more separation off of allosteric, okay? So now we have allosteric stuff that affects the enzyme without affecting the binding site itself, right? Or binding to the binding site. And then we have allosteric compounds. These are getting affected by heterotropic effects, which is the effects of the non-substrate molecules on the allosteric enzymes. So these are going to bind at sites other than your active site again. They're going to shift the Km. They're going to impact the ATCase activity by altering the T and R state ratios. Isozymes are going to provide your means of regulation specific to distinct tissues and developmental stages. You've probably already encountered isozymes to some degree. Have you guys done anything where you monitor like fetal proteins and you had different types of isotopes? No? I've never seen anything like this. Okay. You are aware though, like a mother with her child, right? The child actually changes the types of myoglobin as it ages. Okay, you're kind of aware of that? Okay. That's isozymes. All right. So anyways, you'll see different isozymes in different tissues and different developmental stages of an organism. The isozymes or isoenzymes is also another way of stating that, because apparently we have to skip 2 letters to make it faster. Apparently. These are the enzymes that are going to differ in the amino acid sequence. They'll catalyze the same reactions, though. They're typically going to display different kinetic parameters or respond to different regulatory molecules. They'll be encoded by different genes. These may be expressed in tissue-specific or developmental stage-specific patterns. And so you would typically see they'd have a pattern for expressing, and they may express in the heart, lungs, and then a different one in the tissues. Okay? Or you may see as you age out and get 4 months out, 9 months out from birth, you would have different isozymes start showing up. Okay? And they'll actually have a different in genetic expression. The lactase dehydrogenase isozymes are a really good example of this. The lactate dehydrogenase, LDH, it's going to be a tetrameric protein that's going to catalyze the step in anaerobic glucose metabolism and glucose synthesis. Humans actually have 2 isozymic polypeptide chains for this LDH. We have the H protein that's highly expressed in heart muscle. We have the M protein that's highly expressed in skeletal muscle. And so those are 2 different isozymes that are being expressed in different parts of our bodies, different tissues, right? Okay, so these are the 5 isozymes of lactate dehydrogenase that we're seeing right here. The isozyme proportions will actually change throughout a rat heart development as the tissue switches from the anaerobic to aerobic environment, i.e. as it days before birth, days after birth, right? Okay. The LDH isozyme content is going to also vary by tissue type as well, because again, we have more and less anaerobic versus aerobic environments, don't we? Okay? And we're just optimizing it for those oxygen conditions that we would expect to see. Okay, though not us, unless you're a rat. All right. Now, these are the different LDH isozymes. Is everybody okay with that so far? Okay, they're going to have different amino acid sequences, but very similar catalytic activities. Okay? And they are essentially the same enzyme. They're just different arrangements, different amino acid arrangements of them. Okay, and then this is the days, 0 being birth. Okay, so days before birth, days after birth, and then they become an adult. Okay, and right here you can see that we have the LDH5. These are the subunits, by the way, represented as circular and square. The circular ones here represent the muscle, I believe, M4, if I remember correctly. Yes, I believe it's the M4. Yeah. And then we slowly gain more of these subunits as the H1, right? Can everybody say it okay? Okay. This is the relationship we see as we age. Notice as we get closer and closer to the time of birth, we have a changeover point, and this time of birth is not always perfect, right? And it's what you start seeing is you actually get a bulge of the expression, right? Where you have multiple different types in case. And then you start thinning out a bit to less variance up here. And we have just the 2 forms. Note here where we look at the different tissue types rather than the cycle of life, as it were, that we actually start seeing this, which is, for example, your muscle and liver has more of the M4 that's optimized for more anaerobic conditions, right? And then we have in the heart and lungs areas and red blood cell areas, ones that are a bit more optimized for high oxygen conditions, don't we? Also in the brain, we have a wide variety of them. Why is that? Notice that? Like your kidney, you have a pretty wide variety. Brain, you have a pretty wide variety. White blood cells, you have a wide variety. Okay. So your heart doesn't exactly have a huge variety of aerobic conditions, does it? It's the first place you're going to get any of the blood, right? So you get the oxygen from the lungs, and the heart's pretty much the first one that benefits. Okay? And so high oxygen, right? When we come over to these other cases, your kidneys, your red blood cells, your brain, your red blood cells obviously get these very easily because they're moving with the blood, right? So you pretty much have a pretty consistent amount. But your kidneys and brain are one of the first stops for the blood. However, they aren't the exactly first stop. It's the heart, isn't it? So when you start getting low on oxygen, the kidneys and the brain can actually suffer. Okay? And then you have white blood cells that are all over the place and everywhere, right? All right. So a lot of this stuff actually makes sense when you start thinking about it. This is the covalent modification. This is going to be one of the means.
Why is it section 10.3 before it's set?
Good question.
Well, I had it printed off and from here on out it's 10.4.
If I remember correctly, this used to be in chapter 10.
Okay. I was just wondering to make sure it didn't.
Basically, the previous book before they reorganized. Yeah. No, you're good. Yeah. This year's book has a lot of rearrangement, and a lot of the materials are still that first year after their big rearrangement, and so have not been proofread as well. By the way, anybody want to remind me of this? To change this, you can get a little bit of bonus. Not a lot, but very, very low. Don't forget to send the email. Okay, so the covalent modification, again, this is going to be regulating your enzyme activity. We've already kind of talked about this, but covalent modification is going to be covalent attachment of a modifying functional group to an enzyme. In this case, we see an example of that. Most of these are reversible in terms of we have an enzyme, it will reverse that reaction at some point, okay? And that's because we don't want to have to scrap and remake our enzymes every time, right? But some of the examples of this are going to be phosphorylation, dephosphorylation, acetylation, and deacetylation. Notice these are pairs right here. See that? Also notice that usually the same enzyme will do both ways, but we will have one way that may be preferred due to various things like allosteric interactions, right? Or what other compounds happen to be available. Note again, this is an irreversible modification, as we talked about them before, right? You can't have this just detach on its own with nothing else, okay? But we do have ways of reversing this modification if we do the reaction, right? Did that make sense to everybody? Okay. So some of the common covalent modifications of protein activity are these ones right here. These show you some different examples. This is the modifications right here. These are the donor molecules that you would need to do that. This is the example of a modified protein right here. And then this would be the protein function that's getting modified. Okay? Anybody have any questions about that? No? Okay, fantastic. Okay, so the protein kinases, this is going to catalyze the phosphorylation of the protein substrates by attaching a phosphoryl group. Typically, this is done to a SER, THR, or TYR residue. The ATP is the most common phosphoryl group donor, though not the only one that's possible to use. Okay? But by far and away the most common phosphate donor for any purpose at all. Okay, this is actually the overall reaction here. And so here we have a protein kinase. It's going to take an ATP in this case. We're going to transfer the phosphate to the serine. Actually, we don't know which one this is because we have a... okay, one of those three that would be the ones that we normally attack. Okay. And we're going to get a phosphorylated protein. Now, this phosphorylated protein, I want to break a habit that people get into. Phosphorylated protein doesn't mean activated or inactivated, okay? It can mean either. Some proteins activate when you phosphorylate them. Some of them deactivate, okay? It entirely depends on the protein and whether it should have a phosphate group in that area for it to be an active protein, right? Okay. Okay, notice again, this is an equilibrium. If you left this, it would go backwards and forwards. All right. Okay. Does anybody have any questions? You do get ADP out of this. Okay. Fantastic. Okay, and that's your kinase. This will be a protein of some sort, the protein that works on kinase. Okay? Typically, it would have the name of another protein in it. That's the one it's going to be kinasing, as it were. Okay? And so kinase is involving what again? Using ATP to phosphorylate. Technically, it could also do what? Dephosphorylate, yeah. Because it can go either way, can't it? Okay. Okay, so some of the examples of serine threonine kinases and their activating signals. Only intracellular proteins are going to be regulated by reversible phosphorylation. The examples of serine threonine kinases and their activating signals are these right here. For example, this right here is cyclic nucleotides. This enzyme will then take the cyclic AMP, dependent protein kinase, cyclic GMP, dependent protein kinase. We'll actually give more specific examples of this later, and we'll actually look at it more later in the signaling chapters. But you need to understand the overall, this is overall strategies, right, that we're trying to cover right now. Okay, so consensus sequences. The individual kinases may recognize many related amino acid sequences or consensus sequences. The sequence is not required. So some of the examples of this are protein kinase A. You guys ever heard of protein kinase A yet? In your like cell biology and stuff? Okay. Maybe? Okay. Okay. You very likely have to some degree. But you may have been like pKa or, yeah, you may have been really, really vague about it. Okay, so the protein kinase A will recognize arg something, ser something else. Okay? Or it'll recognize arg something, threonine something else, right? Okay. And so these don't have to be specifically anything. They can be whatever, right? This X right here is a small residue. Z right here can be any large hydrophobic residue, any of those. Okay? And then you can have one of the serine or threonine for the site of the phosphorylation. Now, this does require one of those two because you have to have that OH group to attach, right? Okay. Okay. And that's what that means, consensus sequence. It's a sequence, but it doesn't have to be exacting, right? We have some options. There's only a few spots that have to be exact. Okay, so protein phosphatases. Protein phosphatases are going to catalyze the removal of your phosphoryl groups attached proteins by hydrolyzing the bond, attaching the phosphoryl group. And so a protein phosphatase is going to take a phosphorylated protein. It will then hydrolyze the bond and give you an orthophosphate. Now keep in mind that if we had to have the kinase go backwards, we would have had to have ADP do that reaction, and that reaction will be fairly limited, okay, by those factors. Now this, on the other hand, can just free up a phosphatase. See that? The free phosphate. Specifically an orthophosphate, which is that PI. That's that free one we had talked about. Okay. And so this one can do it really fast, right? Ding, 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 ding. We don't have to wait for ADP to come along. Okay? Okay. Anybody have any questions about that? Okay. So again, protein phosphatases. Are these actually called protein phosphatases, by the way? Or protein kinases? No, you'd have a protein's name, right? This is a class. Okay, so some of the reactions that are catalyzed by kinases and phosphatases are going to be irreversible under cellular conditions. Phosphorylation and dephosphorylation reactions are not the reverse of one another. So when we look at this, I tried to be careful about not using that terminology in that terms. You may have noticed that I was trying not to use phosphorylate and dephosphorylate as direct oppositions of each other. They undo one another, but they're not actually reverses of each other. Okay? You'll notice that they are very different reactions. See that? This is phosphorylation. In this phosphorylated protein, yes, we make a phosphorylated protein. And that's what the product is. But the other product in this is we go from ATP to ADP, right? Those are not in this reaction at all, are they? This is not the products of that previous reaction, is it? No. We have a phosphorylated protein, which is one of the products. But in this case, we have water. And so this is a hydrolysis reaction. They're separate reactions. They're not reverses of each other. It is not actually dephosphorylation if you were to go backwards here. Okay? You just did the reverse reaction for phosphorylation. Okay? Does that make sense? It's a weird terminology thing, but it has to be that way. Yeah. This is dephosphorylation, okay? Okay, so again, they're not reverses of one another. They're different reactions. They have different energies. They have water for one. We have ATP for the other. Okay? These reactions take place at negligible rate in the absence of enzymes. They're very, very slow. And so we can mostly ignore those for physiological purposes when we're talking about them going backwards. And this is because of the free energy here and the activation energies of these. The target proteins will cycle between unphosphorylated and phosphorylated forms. That's what this causes. It causes you to have a protein that it would be unphosphorylated, which are these right here, okay? And it causes you to have a phosphorylated protein, which is this one. Now notice I did not say active or inactive because that depends on the protein, okay? Okay, and this right here is actually the order in terms of free energy. And we can see here that we have the free phosphate here. Here we have the ADP, here we have the ATP, okay? Now, phosphorylation is going to be highly effective means of regulating the activities of target proteins. Phosphorylation is highly effective because the free energy of phosphorylation is very large, meaning it's hard to gather that amount of energy in a normal cellular conditions, okay, without ATP being involved. A phosphoryl group is going to add 2 negative charges to a protein. This will actually potentially alter the electrostatic interactions. I mean, it will alter the electrostatic interactions, period, right? But potentially those are meaningful. Does that make sense? Okay. A phosphoryl group can form 3+ hydrogen bonds. I mean, notice that this group right here has 3 or more hydrogen bond acceptors, doesn't it? And that's for the completely deprotonated one. The kinetics of phosphorylation and dephosphorylation can be adjusted to meet timing needs of particular processes, depending on how much exposure to the enzymes that do them they have, and how many concentrations of ATP or water you have, right? So you can time these to have the correct concentrations at the correct timings, okay? Or in the correct conditions. The phosphorylation can be used to amplify signals because it can be a continuous process where you keep making more and more and more and more signaling molecule, which is the one I would have thought maybe you had seen, but don't worry, we'll get to. Yeah. ATP is going to be your cellular energy currency, and this is one of the things that's involved in this, right? And so it's an energy currency that's widely applicable and used everywhere. It also relates everything to ATP then. So for example, if you want to make it so it's active when you have more ATP, you could. Okay?
Well, we've done where we have hydrolysis reactions or it takes ATP or GTP changes to GDP, which signals or help the signal.
Well, yeah, those signalings, this is going to be part of that. Yeah. And we'll fill that in now. Not now, but so cyclic AMP, this is going to activate the protein kinase A. PKA is how you may have heard it before. This does it by altering the quaternary structure. This is actually the CAMP, the cyclic AMP right here. Okay, can you see the cyclic right there with the phosphate as opposed to AMP, which would be, straight, not cyclic. All right. The epinephrine or adrenaline, which induces the fight or flight response in muscles. Epinephrine actually triggers the formation of the intracellular messenger cyclic AMP, which is this right here. The CAMP is then going to activate the protein kinase A, which is our PKA we just referred to up here. Remember that is that kinase that's going to do that phosphorylation reactions. The PKA is then going to phosphorylate the target proteins to alter activity. And so what we get is we get a cascading effect, right? So we get a small amount of CAMP activating an enzyme. Okay, that's PKA. PKA then is going to sit there activating enzymes to do reactions. Okay? Right. And then those are going to start doing reactions, right? And so we're going to we could have 100 times every step, couldn't we? Yeah. So a little CAMP all of a sudden does a 10,000 times the effect reaction. Okay? And that's why hormones and regulators and the signaling molecules work so well, in case you're wondering. Okay, so the allosteric regulation of the PKA in the absence of CAMP, the regulatory R and catalytic C subunits of the PKA are going to form an inactive R2-C2 complex. This is the inactive version of the PKA. The binding of two CAMP molecules to each of the R units, remember those are the regulatory units, is going to lead to the dissociation of the complex. This is going to be into an R2 subunit and two enzymatically active C subunits. Notice, by the way, the R2 subunit stays together. The C subunits do not stay together. See that? They are two enzymatically active C subunits. Okay. So the PKA is going to exist in isozymic forms. This meets different cellular or developmental needs. PKA is involved in signaling processes throughout your body, and it does it differently depending on whether you're in the liver, kidneys, muscle, heart, brain. Okay? So you're going to see a bunch of these different effects when we get into signaling. But right now we're just talking about the overall strategy it uses. Okay? Okay, so the pseudo-substrate sequence is going to be the sequence that closely matches the consensus sequence. All right, here's our wonderful diagram so we can start understanding these better. So the binding of the CAMP to the regulatory subunits, here's our regulatory subunits again. Everybody see that okay? The little ones look like Pac-Man. And then come in here to this is where they join together into that dimer. Okay. Okay, so they do look like Pac-Man, right? And these are little pills. Come on. It's totally Pac-Man. All right, so the pseudo-substrate sequence of the R subunits is going to occupy the catalytic site of your C subunits until the CAMP binds to the R chains. This is a really great diagram that does a great job on this, by the way. Again, these are our pseudo-substrate sequences. This would be our active sites here that are being bound. This is why we're inactive before we activate these, okay? Because we are actually have part of the protein locking that active site, don't we? Okay. Okay, so we bind to CAMP. This induces a structural change, and that's going to then release the C subunits, which are now active. Notice they're released separately. They don't stay together. They're monomers, okay? They're subunits by themselves. Okay, and we have completely changed the quaternary structure of this enzyme, haven't we? Not a little twist here, a little bend there. We literally release two of the subunits and then have two subunit part left. Okay? It's a very big difference. Is everybody good with that? Does anybody have any issues? Did I lose anybody at all? Fantastic. So this right here is probably one of the most important ones to understand of any of the regulatory stuff if you're going into a medical field. Okay? pKa and how it works is probably one of the single most important processes you would actually deal with. The mutations in the prokinase A can cause Cushing syndrome, for example. Cushing syndrome is a collection of diseases that results from excessive cortisol secretion by the adrenal cortex. Sorry about that. Mm-hmm. This is the one that will bind the substrate right there. Yeah, it will bind substrate. Yeah. So this is the actual active, and this right here is specifically the active site, which would bind substrate and do the reaction. Okay? Yes, the R right here actually has a sequence here that matches up very closely to what our sequence of our substrate would be. Yeah. And so what happens is when we see how it buckles more? Yeah. So when we bind the CAMP, we induce a change in the quaternary structure. This causes that to no longer fit those binding sites properly.
And then that.
Releases, and then we have the binding site for the substrates that are available, and they float off and do the reactions. Mm-hmm. Okay. Yes, so when your CAMP starts lowering, remember this is typically going, this is not covalent modification. This is just bound in there, normal, I forget, but I think it's hydrogen bonding probably. There's so much material, right? Off the top of my head without any preparation, even I might forget some. So this right here, these are going to drift off over time. So basically what happens is you're going to have the signal and it's going to come out, CAMP, okay? Eventually you're going to stop sending that signal. Your CAMP levels will drop and this is going to start having the CAMPs detach. This will then go to that conformation and it'll start binding these active sites as if it was a competitive inhibitor, but it will do it better than a normal competitive inhibitor would. Okay? Does that make more sense? Yeah. Yes. Yep, you got it. Yeah, these are the 4 subunits. This would be the complete assembled quaternary structure. Yeah. This would be the active quaternary structure, which is kind of funny because now we're split into 4 units. Or 3 units, I should say. Yeah. But no problem. No, this is a good one to ask questions on, especially because honestly, if only more people understood pKa better. It really is an important deal. Where was I stop?
You just started it. I stopped before you got into it.
Oh, no problem. I'm like, I don't remember anything. Cushing syndrome. So a collection of diseases that result from excess cortisol secretion by the adrenal cortex. This characterizes symptoms such as muscle weakness, thinning skin that is easily bruised, and osteoporosis. This is commonly caused by mutations within the C subunit that disrupt that interaction between the R and C subunits. And so what this results in is your pKa is activated even in the absence of CAMP. And so you're activating all these damn enzymatic pathways in your different tissues that shouldn't be activated all the time, even without giving off the signal, right? Okay. And so the phosphorylated states of the proteome can be measured. So the phosphoproteome, these are going to be all proteins that are modified by phosphorylation. The phosphoproteomics is going to be the study of phosphoproteomes. I know, right? It has its own field of study. You guys are all suitably oppressed, I guess? Okay. All right, so the exercise is actually going to modify your phosphoproteome, by the way. So exercise will result in the phosphorylation of approximately 1,000 potential phosphorylation sites on approximately 600 different proteins. This is catalyzed by pKa, AMP-activated kinases, AMPK, literally ones that are activated with AMP, right? And other kinases. This is going to alter a number of biological functions, including increasing the ability to process fuel aerobically. By the way, do you know what the single most largest determinant of how long you'll live or how healthy you're going to be is? Exercise. By far and away, period. Okay? Somebody with a hereditary disease that exercises regularly typically lives longer than somebody with no hereditary diseases at all that does not exercise regularly. So it's actually a huge deal. And that's actually one of the newer sciences because you have to do a lot of work monitoring people, right? Takes time. Many enzymes are going to be activated by specific proteolytic cleavages. The folded forms of some enzymes are going to be inactive until one or multiple specific peptide bonds are cleaved. A zymogen. Guys heard of zymogens yet? No? Ooh, I get to introduce you. How about proenzymes? Cool. I got to get your first on this. Okay, so really cool thing, zymogens or proenzymes. From the name proenzyme, by the way, what do you think it's going to be? Pro is usually before, right? Enzyme. Yeah, the inactive precursor to an enzyme. And so it's something that will be an enzyme once you cleave it and make it do something. Okay? And so proteolytic cleavage does not require an energy source. It is irreversible. The proteolytic cleavage is going to play a key role in several biochemical processes. The big one that you probably are going to hopefully jump to right away would have been digestive enzymes, right? So when you digest something, you get a bunch of stuff available, and you need to rapidly release a bunch of enzymes to do it, right? And so what do? Well, you make a bunch of zymogens or proenzymes, you have them ready, and then when you eat something, you'll then release them, okay? And they'll go to work on it so you can rapidly digest it. Anyways, this is going to hydrolyze foodstuffs that are synthesized as zymogens in the stomach and pancreas. Blood clotting is mediated by a cascade of proteolytic activations. The numbering of those proteolytic activations and blood clotting makes no fucking sense, but the one in digestion makes great sense. So sometimes they get that numbering right, sometimes they get it wrong. The one you may actually have known before is some of the protein hormones, because you may have known that due to insulin. So, for example, insulin. These are going to be synthesized as inactive precursors. They'll then be cleaved. The cleaved form is the active form that's going to actually signal, hey, I got a lot of glucose. I need to start, doing something with it. Okay? Okay, so collagen, that's the major constituent of skin and bones. This is actually derived from procollagen, which is a what? This is your calendar, daily calendar. Which is a zymogen or proenzyme, either way. There you go. Zymogens of the stomach and pancreas are some of these right here. So the stomach, one of the main zymogens that you would talk about would be pepsinogen. By the way, notice the zymogen part right here. So pepsin, ogen for zymogen of it. Okay, and then that active enzyme would be called pepsin. The pancreas, we have chymotrypsinogen. And over here we have chymotrypsin, right? And that's the active form. Over here we have the trypsinogen. This is trypsin. Anybody want to try that with me? Trypsinogen. Just basically say the enzyme and then wait a little bit so your tongue resets, and ogen. Okay? Okay, anyways, I think y'all got that okay. So the proteolytic cleavage is going to play a key role in several development processes. For example, collagen is going to be broken down in the mammalian uterus after delivery. This is going to follow the conversions of procollagenase into collagenase. You actually may have noticed this. Like, have you ever seen a pregnant woman before and after the pregnancy? So you may notice that, like, before the pregnancy, her hair starts fading and becoming less shiny and stuff, and her skin also. And then after the pregnancy, typically they're known like, you have the glow. And her hair like becomes shinier and stuff. That's collagen, right? Yeah. She's incorporating the collagen from after birth. So apoptosis, or programmed cell death. This is going to sculpt shapes or the body parts during development, and it will eliminate the damaged or infected cells. This is actually mediated by capaces, which is synthesized as procapaces. Okay, everybody knows apoptosis, right? Yeah, okay. There's some parts here where I'm like, I'm working on top of the biology, and I'm not sure if you've gotten there on the biology. So please let me know if I'm working on scaffolding that isn't there. Okay, the chymotrypsin. This is a digestive enzyme that's going to cleave the peptide bonds on the carboxyl side of the amino acid residues with large hydrophobic R groups. Yes, that same chymotrypsin that we've talked about, 3-4 times now? Same one, okay? That's going to happen a lot because we're looking at these from different parts of our perspectives, right? And we're building up a framework for you to understand biochemistry through, okay? Okay, so these are synthesized as an inactive precursor. These are called the chymotrypsinogens that we talked about before. This is going to happen in the acinar cells of the pancreas. The zymogens are going to be synthesized again. Many of the zymogens are going to be synthesized, especially in the pancreas specifically, in the acinar cells, where they will then secrete the zymogens. And you may have actually seen this figure before in other books. Have you seen that before? No? Oh, did you? Okay, cool. Good timing. Anyways, zymogens, synthesized in the acinar cells of the pancreas, stored inside the membrane-bound granules, and here's the zymogen granule here, right? Okay. And then this is going to be released into the duodenum when the cell receives a hormone signal or a nerve impulse. And so right here, we have the ribosomes attached to the endoplasmic reticulum. We're synthesizing. They're going to get transported to the Golgi complex. We're going to assemble. We'll get our zymogen granule. So we have lots of zymogens available in our own little pills, don't we? See that? Okay. This is our strategy to overcome the blood-brain barrier, as it were. Okay? The hydrophilic barrier. And so we're going to take those zymogens. Those are going to be released out into the lumen. They can also be released into different areas, but in this case, we're talking about the pancreas. So we're looking at the ER complexes. So yeah. You can do the same thing with myosins. They're just bigger versions of this. And they'd have more than just the zymogens. They'd have a bunch of stuff. Okay? Okay, so proteolysis activates the chymotrypsinogen. The specific cleavage of the peptide bond is going to be the joining ARG, I have to say it, ARG15, and I'm not going to do it. 16. IL 16. This is the trypsin is going to yield the active enzyme. This will be this form of the chymotrypsin, okay? The pi form of chymotrypsin right here. Okay, so here we have the inactive form of the chymotrypsinogen, right? The inactive form, the zymogen, okay? And what we're going to do is we're then going to have trypsin. It's actually going to cleave this connection right here, which is right there. See that cleavage? Okay. And then that's going to form the pi chymotrypsin, which is an active form of chymotrypsin. However, we can have two subsequent cleavages that will happen due to the pi chymotrypsin will actually cleave other pi chymotrypsins. Okay? And this will actually give us two dipeptides here. It also is going to give us the stable enzyme, which is the alpha chymotrypsin, which is another active form, but this active form is going to do the total results we wanted out of the chymotrypsin. Okay? Not just cleave itself. Right. Did that make sense? Okay. And that's why we can get a zymogen burst, as it were. Okay? Or an enzyme burst. We get a bunch of active enzyme all in one place, all at one time. Okay? And have really rapid digestion. Okay, this is the C chain right here, by the way. B chain, A chain. Does anybody have any questions about that? Fantastic. Please note that this does wrap around on itself. This is not a straight chain going off here. Okay? Okay. This is going to lead to the formation of our substrate binding site. Notice that before we do this, we actually do not have an open substrate binding site, do we? No, it's actually contained and blocked in there. Okay? It's only after we do these cleavages that we get substrate binding sites. Ironically, gamma chymotrypsin substrate binding site is for gamma chymotrypsin itself, right? But alpha chymotrypsin has a substrate that is available to do a bunch of reactions. Well, a specific cleavage of a bunch of different proteins, right? Okay, so the cleavage of the peptide bond joining the 15th and 16th amino acids is going to trigger the key conformational changes that's going to lead to opening that substrate site. The 16th one is actually going to form an ionic bond with ASP194. This is going to trigger our formation of our substrate-specific pocket. This is why we literally get a conformational change to make a new substrate pocket that we can then use. Okay? The ASP194 is going to repositioning is going to rearrange the Gly195. This is going to enable the proper formation of our oxyanion hole. You remember your catalytic triad, oxyanion hole, the chymotrypsin, where we talked about that stuff? That's this. That binding site that we were like going over and we did all those diagrams for and we looked at and oh, that's so cool. Well, that wasn't even formed in the zymogen. We actually had to cleave first. Okay? Okay. And so this is actually what it looks like. This is the zymogen right here. Can everybody see it okay? And you see actually that these are not available to do the oxyanion hole. See, it's out in the wrong shape and the wrong orientation, isn't it? Okay? And this right here is not available to do any of the reactions or anything. We can even see that this is a different orientation for this site. And then the trypsin is going to work on the chymotrypsin. This is going to make our oxyanion hole once it has that new binding arrangement. And we can start doing reactions. So the generation of trypsin from trypsinogen is going to lead to your activation of your other zymogens. The trypsinogen is actually going to be converted to the active trypsin through by enteropeptidase. So the trypsin is going to activate trypsinogen and other zymogens. This is actually the activation cascade. Okay, remember this is a cascade because it's going to give you a burst. Okay, anybody ever seen like the in Gen Chem 2? Did anybody go over like what makes a nuclear bomb be a nuclear bomb? No? Okay. You know chain reactions, right? So chain reactions always require you to make more than, so if you do one thing into one thing to one thing to one thing, it never increases, right? You always end up with one thing at the end. Okay? If you do one thing to two things, one thing to two things, one thing to two things, always repeatedly, what's going to happen? you're going to grow exponentially, right? And same thing with 3, 4, 5. So if you do that with an enzyme, each enzyme could do hundreds of these cycles, right? And what that means is that each step of these could be a factor of 100 multiplication, right? So 100, 1,000, 10,000, a million, and we'll just keep getting a bigger and bigger burst every step, won't we? Okay? And that's why enzyme cascades happen. They're basically where we want to get a really big burst of activity all in one place at one time. Okay? Okay, so again, the enteropeptidase is going to act on the trypsin. This is going to go from the trypsinogen to the trypsin. Note that the trypsin will actually come back and start acting on itself. Okay? The trypsin is then also going to act on the trypsinogen, the proteolases, the procarboxylpeptidase, the prolipase, and all of those. And what these are then going to do is they're going to do all those reactions that we associate with the digestion processes. Okay? And that's how we get that huge burst right away. Okay, so some proteolytic enzymes have specific inhibitors. So the serpins, what a great abbreviation, right? The serpins. Anyways, serine protease inhibitors, that's the abbreviation that is, in case you get confused. SER, that's the protease P inhibitor, okay? And that's going to be a specific type of protease inhibitor. We have the pancreatic trypsin, which is going to be the inhibitor that inhibits trypsin by binding very tightly to trypsin's active site. So this right here is the trypsin inhibitor, the pancreatic-free pancreatic trypsin inhibitor right there. See, it actually has the binding site sequence right there that's going to bind to that. It actually comes in here. This is then the pancreatic trypsin inhibitor that's actually bound to trypsin and is now blocking that active site. Okay? What type of inhibitor is that, by the way? Mm-hmm. You got it. Competitive. Okay, so the function of proteolytic inhibitors, the trypsin inhibitor is going to prevent traces of prematurely activated trypsin from initiating that cascade, where the zymogens are still in the pancreas or the pancreatic ducts. It's going to prevent severe damage to tissues. Remember, we're also made out of that same stuff we would be digesting, right? There's not that much difference between us and a cow, right? So what if we did this burst right here in one of our cells and we have nothing else to digest? We're digesting our cell, aren't we? Yeah, we have a problem. And if we have nothing to turn off the burst, what do we do? We'll just keep digesting ourselves, won't we? Okay? And so this is actually the reason why this works so well as the competitive inhibitor, right? So if we keep a moderate amount of this around our cells at all time, if one of these accidentally activates, what happens? It gets inhibited, doesn't it? Okay? But what if it's now exposed to a bunch of stuff it should be digesting, like a bunch of zymogens, because we just did a burst? Well, then the competitive inhibitor is going to be overwhelmed, isn't it? Okay? Is it starting to come together? Yeah. Okay. So the alpha-1 antitrypsin is going to protect tissues from digestion by the elastase. The alpha-1 antitrypsin deficiency is actually going to cause excess elastase. There's actually just genetic disorders that lead to the deficiency of the alpha-1 antitrypsin. This will cause excess lactase. The damages to the alveolo. God, let me get some water. Oh, my Thursdays. It's a bit much. Damages to the alveolar wall by digesting connective tissue proteins. You're digesting your own proteins again. Results in Emphysema. Emphysema. God. Okay, emphysema. Good grief. La, right? I need to do some exercises on that. Now, the cigarette smoke can actually induce the oxidation of methionine. So smoking cigarettes actually greatly increases your chance of an individual with a genetic disorder will develop emphysema. And this is why it increases the risk of whether you're going to get emphysema from smoking cigarettes, okay? Because you do have to be genetically predisposed towards this for it to happen. The smoke is going to oxidize the MET358 of the inhibitor to methionine sulfoxide. That's methionine sulfoxide, by the way. The unmodified MET358 is going to be essential for binding the elastase. And so we've essentially covalently modified this to no longer be able to inhibit that, right? And so what's going to end up happening is we're going to end up digesting ourselves a little. Okay? And you know, smoking is so wonderful, right? Just have that picture in your head, huh? So enzymatic cascades are going to allow for rapid responses. Now we're going to start looking at blood clotting, okay? And so the enzymatic cascade is going to be, again, that series of zymogen activations. This will amplify the signal at each step. This is used to achieve a rapid response and a drastic rapid response. The hemostasis, this is going to be the process of blood clot formation and dissolution. So there's two pathways to initiate blood clotting. There's the intrinsic pathway. This is going to be the one that's activated by exposure of anionic surfaces upon rupture of the endothelial lining of the blood vessels. There's the extrinsic, Jesus. Man, I need something to. There's the extrinsic pathway that's initiated when trauma exposes tissue factor being abbreviated TF, of an integral membrane glycoprotein. Now, this one actually here, the extrinsic pathway, is the one that most of the time is the real big one we focus on, okay? And that appears to be the most crucial in blood clotting, though there is the intrinsic pathway. It does exist, it happens, and so you need to be aware of it, okay? The blood clotting cascade steps, both pathways are going to activate the serine protease factor X, or 10 actually, it's not actually an X. Greek, Latin numbering 10X. Okay. Okay. Factor X, as it were, or 10, converts the zymogen prothrombin into a thrombin. The thrombin is going to amplify the clotting process through a positive feedback that's going to generate more thrombin. So remember, the thrombin is going to circle back around and start activating more of the prothrombins, okay? And this will then cascade and create a larger and larger effect on that step, which will create larger effects on the next step, right? And so thrombin is going to convert fibrinogen into fibrin clots. Now, when you are clotting, a good way of thinking about that is you're doing, you know how we talked about the fibers where they're cross-linking in organic chemistry a little bit? Okay, think of it like that. That's what you're doing. You're growing polymer fibers and then cross-linking them to patch it up, okay? And that's your clot. This is actually the cascade right here. And you can see here's the intrinsic pathway. This is going to be linked to a damaged surface. This right here is the extrinsic pathway. Now remember, the extrinsic pathway is typically the one that's the most common. The ones that's usually the most crucial in blood clotting. It's the one that, if you're going to remember one of these and not the other, at least remember the extrinsic, okay? The intrinsic, on the other way, is due to the exposure of the surfaces upon rupture of the endothelial lining of your blood vessels, which is right here. This is that damaged surface right here. Okay, so the active forms of your clotting factors in each of these is going to be denoted with an A, that subscript A. Can everybody see it okay? In the yellow boxes here and blue box here. So can everybody see the A's okay? Okay. Okay. Each of these would be factor, this one's 12, right? This one's 11, this one's 9. See that? Okay. So the factors that are enzymes or enzyme cofactors are going to be activated by thrombin, are going to be designated with an asterisk. So we look at this asterisk here, and what does that asterisk mean? Activated by thrombin. You got it. Yeah. Notice those are kind of key steps. No, down here as well. And up here as well. Okay. There's a bigger one in your textbook that's really easy to look at. And you can also just grab this slide and zoom it. You got it. You got it. It's also down here. Yeah. It's easier just to look at the figure for those. It shows up in a number of places because thrombin is probably the most important part of this. Like once you get thrombin going, it's actually hard to not experience some clotting. Okay? You may not get the full clotting effect, but once thrombin's going, you're getting something. You're definitely getting something. Okay, so this is the full pathway. Again, this is the extrinsic pathway. You'll have factor 7 going to factor 7, the activated form. Note, by the way, the thrombin will actually act on this one right here. Okay? Now, this is going to then have, along with tissue factor, that's that TF. Remember how that showed up in the extrinsic definition? TF, tissue factor. Okay. This is actually going to be typically released by your trauma. Along with the tissue factor, factor 7 is actually going to work on factor 10. Factor 10 is going to be activated. The activated form of factor 10 along with factor 5, the activated form of factor 5, is going to then take prothrombin. It's going to convert it to thrombin. Thrombin, by the way, is also called factor what? Factor 2, right? Yeah, it's factor 2. Okay? So keep that in mind. Yes, thrombin is thrombin, but it's also called factor 2 here.
So for the extrinsic, the tissue factor causes factor 10?
You need both. You need factor 7 and tissue factor. And then that's going to cause the factor 10 to be activated. Now keep in mind, factor 7 will actually get activated more of that by thrombin. So we actually have a wraparound that comes back here.
So does the factor 7 activate
It does.
tissue factor?
No, it doesn't activate the tissue factor. The tissue factor is kind of like a coenzyme. Okay, you need both of them. They then activate factor 10. And then once factor 10 is activated, it actually needs to be alongside factor 5. So you need the active forms of 10 and 5, and those then activate the thrombin. Keep in mind that if you did this in the middle of your blood vessel, you would clot your blood vessel, right? Or your heart, you would make a clot in your heart. Okay? So there's a little bit of security here. That is, these two together, yep, work on factor 2, or prothrombin, and make it the active factor 2, which would be thrombin. Okay. By the way, this is the start where you have thrombin, going prothrombin to thrombin. This is the start of your final common pathway. So both of the pathways have merged at this point, right? This thrombin is actually going to activate on a lot of stuff, but directly it actually is going to work on the fibrinogen here, giving you fibrin. That is factor 1, the active form of factor 1. The active form of factor 1 will actually need the factor 13 active form to do the cross-linking of the fibrin. Okay? So if you just had fibrin, you get, you've all have had it where you only have one side and then the cross-link has come out on like a shirt or something and all the different parts come across all over the place, right? That's what it would be like. So if you just had fibrin, it would just be a bunch of fibers all laid out in an area. They wouldn't have any cross-linking or very minimal. And you might get a little tangling, so you get a little bit of clotting, right? But overall, it's not going to clot well, is it? Yeah. They're going to tend to spread and gap and shift everywhere, right? And so the factor 13 is going to act with the fibrin to cross-link the fibrin. That will give you your full clotting effect. Okay. The intrinsic pathway, on the other hand, is a damaged surface effect. That is not necessarily a damaged your surface. That is a damaged surface of a tissue or cell, right? Which is not exactly the same thing. Does that make sense? Because those are inside you? Yes. Yeah. It just does, it's not in the cascade itself. Okay? But it is activated by thrombin down here. Okay, so this is going to be the kinogen kallikrein effect. This is right here, the factor 12. We'll have the activation of factor 12. That's going to give us the activation right here of factor 11. That will give us the activation of factor 9. That then needs to go along with factor 8, the activated form of factor 8, to activate factor 10. Okay. And I'll clarify a bit more of this along these next slides, okay? It's a lot to unload in a single diagram, okay? I know. Okay, so let's talk about the thrombin real quick. This is going to contain 4 distinct protein domains. Our prothrombin will contain these 4 domains, which is the GLA domain, GLA domain. This is rich in the calcium binding gamma carboxyl glutamate, or GLA residues, which is where it takes its name, right? GLA, glutamate, GLA domain. We also have here 2 Kringle domains right there. Those are the Kringle domains, the red ones. This is going to help maintain thrombin into the inactive form. This right here is then the cleavage sites. Remember we were talking about cleaves at those 2 sites? There's your cleavage sites. Okay. This then is the serine protease itself. That's why the cleavage activates that serine protease, right? Okay. So what we get here is the thrombin. This has to bind to calcium to actually be converted to thrombin. And so the binding of the calcium to the GLA domains will anchor the zymogen to the platelets after injury. This actually brings the thrombin close to the factor 10 and factor 5 that is going to catalyze its conversion into thrombin. So that's up here. See those? Right here. And thrombin. Okay? And that process with the calcium anchoring it into the same area is why these 3 things can assemble and actually do stuff. Because otherwise 3 things running together in blood is not exactly the most common event, right? Okay. So right here we have the fibrinogen. This can be converted by thrombin into the fibrin clot. The fibrinogen is denoted by A alpha 2, B beta 2, gamma 2. This gives you the large blood plasma glycoprotein. This is composed of 3 globular regions connected by 2 rods. This is what it looks like right here. Okay? And so this is the gamma globular unit right here. And it's on both sides. Can you see that okay? See the gamma 2, gamma globulars on each side. There's little endings. This right here is the beta globular unit, but there's 2 versions of this. See that? That you end up with 2 versions. We have the B beta globular units. This then is the central globular unit right here, and these are the rods. Everybody okay with that so far? Fantastic. This is what the fibrogen looks like in abstract, so you don't have to have such a strange looking thing to look at. Okay? And you can kind of see a bit more detail about it, right? Okay, so when we look at an abstract, we're going to use that to look at how these would actually link up and make their cross linkages. Okay? And so the thrombin is going to cleave the fibrogen. This is going to yield our A and B peptides, which are our fibrinopeptides, and a fibrin monomer. Okay, this right here is the fibrin monomer right there. A little bit, right? Notice the difference here, which these are the fibrinopeptides. They were the little caps, okay? And that was on that central unit right here. Okay? See, those are the little caps there. Denoted by little circles, right? Okay. So we cleave those off by fibrin. This is then going to give us active sites that we can do polymerization at. Okay? And so what's going to happen here is, again, we'll have the alpha and beta peptides. See how there's 2 different versions of those? I'm sorry, A and B peptides. They were the ones that are attached to the alpha and beta. That's why they're A and B. Okay. And they give us 4 total, 2 of each, right? Okay, and so we have the fibrin monomer. This is the activated fibrin monomer. We're ready to do some reactivity, right? Notice again, we have the alpha, beta. See those? Different coloring. And then we have this right here with the 2 gamma units at the end. Okay, so the next thing that's going to happen is we're going to start reacting. The gamma units will actually attach to these alpha beta units here. Okay? And how they'll attach is you'll have 2 gamma units attaching to each alpha and each beta. And that's why it gives you a polymer, because 2 attach to every one, right? Okay, so these are the alpha and beta units right here, right? Technically, one of them's probably color related, but I just don't care. It doesn't matter. For our purposes, it's basically symmetric. Now, this right here, this would be the gamma units. So one fibrin will attach to 2 other fibrins on one side of it, okay? For a total of 4 fibrins attached to every one. And these are attached by the 1 gamma unit attaching to one side of the alpha unit here, and then a different gamma unit on a different fibrin attaching to that as well on the other side. And that's why it actually ends up being a polymerization, because you get a 1 to 2 attachment every time, and it extends out for basically forever, right? This also is going to give you this, where it's going to assemble multi-layer fibers, right? Can you see that okay? Okay. So here we have 3 layers of the fiber, giving you stronger and stronger fibers each time as it develops. You can actually see this, by the way. Have you ever sort of watched your wound enough that you can see it sort of thickening out over time? Yeah, that's what you're seeing. And you can actually see the in-between stages where it's real thin and fibrous, and then it sort of fills in over time, right? It becomes really hard. Okay? Yeah, that's what we're looking at, isn't it? Okay. Okay, so this is going to be the cross-linking of the fibrin monomers. The fibrin monomers would polymerize. They would form a soft clot at first. This is actually going to be stabilized by cross-linking, and this is through cross-linking at the Lys and Glyn, LYS. Do I have to say out the amino acids each time, or are you okay with me just pointing? Okay, yeah. It takes GLN residues by the transglutamase. This is factor 13, by the way. If you go back up here, 13. Okay, that's at the bottom here. Is there any reason you don't all just like line up in a straight line, by the way? Oh, okay. Yeah, if you ever want, we can bring in a cord. Okay, so anyways, factor 13. That's where we're at right now. We're cross-linking the fibrin. This is actually the reaction that we cross-link the fibrin. If you were to actually state the enzyme's name, it's the transglutaminase. Glutaminase. God, I'm just having a hell of a time today. Literally, glutamine, lysine, linking them along that amide, right? So you'll make a mid linkage. So the vitamin K is actually going to be required for the formation of the gamma carboxyl glutamate. It's actually essential for the synthesis of prothrombin and other clotting factors, which is why if you ever have heard like, you need to get more vitamin K if you're having trouble healing wounds. Okay? Or if you're bleeding too much and you're having blood clotting problems, vitamin K, right? Okay. That's why. The vitamin K, again, required for the synthesis of the gamma carboxyl glutamate by the gamma glutamyl carboxylase. The gamma glutamyl carboxylase is going to require your vitamin K to convert the glue residues in prothrombin to the gamma carboxyl glutamate. And again, this is over here. This is the vitamin K, by the way. The other name for that is quinone, if you're curious. So quinone. The gamma carboxyl glutamate is actually going to be required for the prothrombin activation. So what will happen is your vitamin K here is going to go to the reduced vitamin K, which is the hydroquinone right here. You can see the carbonyls have been reduced to the OHs, the hydroxy groups. This is then your glutamate residue. It's going to react with the gamma glutamyl carboxylase, being the enzyme here. In the presence of CO2 and O2, this will give you this one right here, which is the gamma carboxyl glutamate residue. Interestingly enough, this right here is actually your vitamin K that's been converted into an epoxide. You guys see that okay? Remember those epoxidation reactions of double bonds? You didn't think that was going to show up, did you? Yeah, there you go. Okay, now, again, that epoxide, there's actually enzymes that have a warfarin inhibition right here. These are the proposed sites of warfarin inhibition. These would actually make the vitamin K not be able to go back into the one that would actually do the reaction. Okay? And these are the enzymes that would bring it backwards from the epoxide. Okay, so let's look at some anticoagulant drugs. Again, what's an anticoagulant again? Won't let it clog. You got it. So it's going to be medicines that more specifically reduce blood clot formation. They're going to do things like if you're having clotting problems, they're going to help you prevent strokes, myocardial infarction, pulmonary embolism, and in patients with increased clotting risk. Interestingly enough, this is actually how my father died. They could not use this because he had a bleed. Well, so he threw a clot, hit his head when he fell, and he started leaking blood in his head. And so you can't do anything. Anyways, so an example of this would be obviously warfarin, which is why we brought this example up right here. Okay. And this inhibits the enzymes that are required to regenerate the active form of vitamin K. And so whatever amount of vitamin K you happen to have, it's not going to be enough because you'll continuously use it and then not be able to go back and reset it. Okay? There we go. Note, by the way, this would be quinone reductase right here, and this is the epoxide reductase. So if you get rid of the epoxide off of this, what does this look like? The quinone, right? Okay, and this is warfarin, by the way. What do you notice about warfarin and vitamin K? They have similar structures. So if you were to just guess, you could actually come up with some hypotheses of how this would interact, right? Okay, so the clotting process, this must be precisely regulated. Clotting is going to be limited to the injury site. It's going to have numerous mechanisms to try to prevent this, and yet it still has problems. And yet, right? So, some of the factors are going to be unstable and lose activity fairly quickly. And so when your blood tries to wash them away to the rest of your body, what happens to them? Well, they come apart and stop working, don't they? Okay? And so they only have a relatively local effect, and then they quickly degrade. The clotting factors are going to be diluted by blood flow, and this is going to be removed by the liver, and then degraded by proteases. And so some of the factors will actually wash through with your blood, but they'll be too dilute to have a huge effect, right? And so are they going to be causing clotting in that part of your blood? Yeah, but very slowly, okay? Hopefully slowly enough they can get to your liver, and then they can actually get broken down, degraded, right? Specific inhibitors of clotting factors are going to terminate the clotting. This is the other part that helps you out. You keep a low level of inhibitors to inhibit clotting available, and this is going to make it so clotting has to overcome that inhibition first, right? And usually it does, and it's not a problem because we have a burst, don't we? So in the area where you have clotting and you want clotting, you'll have a big burst. It'll overcome any inhibition. In other areas, it's not going to overcome the inhibition, hopefully. The anticoagulant heparin is going to increase the rate of formation of irreversible complexes between the antithrombin 3 and serine proteases, and the serine proteases of your clotting factors. Okay, ooh, I finished it. Okay, wow. That's actually